Well, I'd first like to say this is a very special occasion for me and to thank Gordon for that lovely introduction. I also would like to say that the Microbiology Society has always been my society. I confess to having been a member for about 40 years, and it really has helped shape my career over this period, and lots of the fantastic things that have come out of work that we've done has been a consequence of collaborations and of meeting people in Microbiology Society meetings. So I really do love the Microbiology Society. So, what am I going to tell you about? Well, I thought I might start off telling you a little about another conference, a much better conference than a Microbiology Society meeting. This is because it was a centenary meeting. It was about molecular biology and infection, and it was the centenary of the establishment of the Institut Pasteur. Now, why am I telling you this? Well, there was this young guy, i.e. me, who had just got his first academic job. And I went down to the pigeonholes. There were no emails in those days, and I found a letter from the Institut Pasteur. And that letter said, "We'd like to invite you to give a plenary lecture at our centenary colloquium." And I thought, what's a plenary lecture? Well, I did do, because I'd been to a Microbiology Society meeting. And of course I said yes. So they wrote back and said, "That's great. We would like you to bring your wife." And I thought, well, Microbiology Societies don't allow wives to come along in this way. And they said, well, it's a very special colloquium because it's going to be opened by the President of France, François Mitterrand, closed by the Prime Minister, Jacques Chirac. The Queen Sophia of Spain is coming to look after the conference, the concert at the Opera House. And we'd like you to come to all those events. Your wife will have a special social program for her. And there'll be 8 Nobel Prize winners and a few other leading people in that field. Well, I think I was about 28, 29 at that point. I did my PhD about 3 years before. So I didn't question it when they said, could you please fly with Air France because the captain will welcome you on board. I thought, this doesn't happen in Microbiology Society meetings. And when we got to Paris, we had a limousine that drove us down the Champs-Élysées to this fantastic hotel. There were even presents on the bed. Again, I didn't get any this time. Might be getting a medal, but they weren't there. And so we had a wonderful time. It was a great meeting, and well, we went home very happy, thinking if this is what French Microbiology Society meetings are like, maybe we could have more of those like that. And it wasn't until a few years later when I bumped into the new director of the Institut Pasteur. And we had a chat about the centenary meeting, how wonderful it was. And Philippe said to me, he said, "Paul, did we ever tell you we made a mistake? We didn't intend to invite you at all. In fact, it was Peter Williams at Leicester who'd done some wonderful work on plasmid-mediated virulence." I said, "Well, thanks, Philippe. We had a wonderful time." So that's how things started off. They do get better, I promise you. One of the Nobel Prize winners at the meeting was Francis Jacob, who you might remember with Andrei Lvov, got the Nobel Prize for working on the LAC operon. And he had some interesting things to say about bacteria. In fact, he thinks that the bacterial world, in fact, he wrote this, is pretty boring. You know, it's peopled by, cells that live without sex, without hormones, without nervous systems. This is all pretty, boring. They don't need to talk to each other. But of course now, 30, 40 years later, we know rather different, that bacteria are able to interact in a very sophisticated way, and they are well capable of behaving collectively. And that's what bacterial cell-cell communication is all about. But I thought what I would do is to start off telling you the science behind the story rather than just telling you the science itself. So I mentioned that in my early 20s, I got a lectureship at Nottingham University. You'll be aware of I'm sure, this kind of graph, which tells you all about the wonderful time in the sort of 30s to the 60s, where lots of new antibiotics were discovered, and then it all dried up. And of course, antibiotics were relatively cheap because many of them were made by fermentation using building blocks like 6-APA and 7-ACA. which you could semi-synthetically alter to actually come up with compounds that resisted beta-lactamases and were much more potent. Now, Barry Bancroft recruited me because he wanted a pet microbiologist, because he was interested in carbapenem antibiotics. These lack the thiazolidine and sulfur, they have a carbon instead. Imipenem was the first one that was in the clinic. And Barry was interested in whether there was somewhere out there the possibility of creating a 6-APA or 7-ACA analog that you could make lots of novel carbapenems, lots of new antibiotics, lots of money for research. And he'd started off doing a little bit of work with classical biochemical radioactive feeding experiments. And the carbapenem nucleus that's made by a soil bacterium, Erwinia carotovera, there's also a serraceous strain that makes this. And Barry had worked out that we needed acetate and glutamate to make a carbapenem, which lacks its double bond here in that ring. And it has the wrong stereochemistry for the antibiotic. It has to change its stereochemistry here and then be desaturated to make this carbapenem. So Barry thought, let's get a microbiologist, because he was a chemist, to suggest some experiments we could do to find out more about this biochemical pathway. So what we did, we didn't have any good genetic tools then, was to actually use chemical mutagenesis to mutagenize the Erwinia producing strain and to come up with some mutants that didn't produce the antibiotic, but when you basically plated them out next to each other, they actually cross-fed each other. So a group 1 mutant made something that a group 2 mutant could take up and use to make the antibiotic. Therefore, we were hoping to find additional intermediates in the pathway and hopefully a way towards getting at the genes. So we started off, as you do, with these mutants. We took the mutant, the group 2 mutant that's producing something that the group 1 is taking up. We grew up about 20 liters of this stuff. Didn't have very good analytical tools in those days. Did some HPLC, bioassayed them to look for supernatants that contained antibiotic activity on this sensitive E. coli, and then had to do some NMR mass spec and chemical synthesis, and found that we had nothing that looked like a carbapenem. It was this wretched molecule here, 3-oxyhexanyl homoserine lactone. Well, we had no idea what that was. We had some great people who did all this work, though. Mavis did all the separation work. She's one of our technicians. She was a fabulous separation scientist. And Ram Chabra, who worked with us for over 30 years, who did all the chemistry. Now, we thought, well, we'll explore this molecule a bit further, see what it does. If we take a group 2 mutant and we give it this 3-oxyhexanyl homoserine lactone, we can get a nice dose-response curve. So it looks, we've made a specific molecule. It looks like this is true. And then we added it to the wild type. So here's your standard growth curve. Production of the carbapenem was density dependent, classic secondary metabolite. If we added our signal molecule, bang, it came on straight away. So it looks like we had a kosher molecule. This was doing something really quite interesting. So as you do with a chemistry group, they made lots and lots of analogs of this molecule, and we figured out again it was very highly specific. Whatever we did to the ring or the side chain, we lost activity. But when Ram went along to the library, again, we didn't have an internet in those days, he went to find this molecule. He came back and said, well, it's not very interesting. He said, it's not going to help us with the carbapenems. It's something to do with, as I'm sure you know, the bob-tailed squid. In fact, something to do with bioluminescence, because in the light organ of that squid, you have a microbe called Vibrio fischeri, which uses the acyl homoserine lactone to make light. And of course, in those days, they'd already worked out the genetics of the system. And so we know we have a transcriptional regulator, LuxR. We have a synthase for the lactone, LuxI. And we have an autoinduction system. So the first name given to this type of molecule was that it was an autoinducer of bioluminescence. LuxC, D, A, B, and E were fatty acid reductase and luciferase, respectively. So that was kind of, well, where do we go next? Well, this is another interesting little story, is that I went along to the inaugural lecture of a new professor at Nottingham who was talking about serendipity in the discovery of antibiotics. And I met someone there called Gordon Stewart. Gordon was in food sciences and microbiology, And Gordon had been playing with bacterial luciferase systems, so he knew all about the lux operon, and he was using it as a system for testing antimicrobials in foods. And Gordon said, do you realize you've actually found something that's quite significant? Because Vibrio fischeri is a marine organism, makes this lactone to control bioluminescence. Erwinia is a soil plant microorganism, uses it, the same molecule, for controlling carbapenem production. Other bacteria must surely do this, and I've got just the bioassay for you. And Gordon had built a biosensor in which used all the lux genes except luxi, just the luxi promoter. So if you added 3-oxo-C6 to this particular biosensor, it lit up, and you could pick that up on X-ray film or with the photo and video camera or luminometer. So we thought, right, well, do other bacteria make this lactone? We could screen culture supernatant, so we went around the lab collecting all the bugs everybody had to throw them into this system. And we also realized we'd use it to clone any luxi-type synthases that might be present in any of those bacteria. So what we now know is that many different bacteria make these acyl homoserine lactones. In fact, there are lots of different luxi synthases. There are also 2 other families of AHL synthases. But we cloned 3 or 4 of these using that genetic complementation approach. And we now know, of course, that the 3-oxo-C6 is only one type of homoserine lactone that is made. The ring is constant, but the side chain changes. You can change the substituent at the 3 position or the length of the chain. So there are lots of different words in this communication language, if you like. And of course, when we go back to the Erwinia story, to carbapenem biosynthesis, and here I should bring in another very valued colleague, and that's George Salmond, because George, unlike us, was very good at Erwinia genetics. And George had developed some really good tools that enabled us to find, or rather his group to find, because he did most of the work, on carbapenem biosynthesis, and also the whole of this CAR operon, which also contained an interesting resistance mechanism, so the producer cell didn't kill itself. So that was the start of our kind of unexpected way into what's become known as quorum sensing. I'm going to digress just a little bit here. Sticking with the antimicrobials theme, there's a very nice purple microbe called Chromobacterium violaceum. And it makes this purple pigment, which actually has antimicrobial properties. And we found that this was regulated by C6, so there's no 3-oxo-oxygen there with this molecule. Now, the reason I mention this initially is because it turned out to be a very valuable bioassay strain. You could streak out the mutant, double mutant that we made. I won't go into the details of that. But we could streak out CVO26 on one way in a plate, and then alongside it, put a test strain. If that test strain made a homoserine lactone, it turned purple. So you didn't need anything sophisticated to find out if your pet microbe made that molecule. You can also use it in bioassay plates, so this is a negative result, these are positive results, and you can overlay plates with thin layer chromatography plates with CVO26 and separate out lots of different signal molecules at once. And some bacteria certainly made lots of different analogues of those homoserine lactones. And of course, there's an I gene, there's an R gene, and there's an interesting repressor, and all this regulates not just the violacing cluster, but also the chitinase and protease. Now, why am I mentioning this particular little story in my scientific journey? Well, that's because this paper came out in Microbiology. It's a very important journal, and I'd like to just emphasize why I think society journals matter. So, this paper has had well over 2,000 citations. So publishing in microbiology doesn't mean everybody will ignore it. This one certainly gets picked up. So why should you publish in society journals? Well, a lot of the funding that comes from publications goes back into supporting members, serving the community. We've got great editors. We're not driven by market forces. The reviewing process, the integrity of that is intact, because we know that in a lot of journals these days, particularly the predatory ones, there are all sorts of issues about the fidelity of work that's being published, particularly now that we've got AI causing additional problems in that field. And of course, whilst the society would like to make profits, it really avoids making excessive profits because everything's plowed back into us and into what we do. And I'd just like to highlight one figure that I spotted in an MBio paper that came out a few years back. And this is publication. So In society journals, publications aren't really climbing very much. But in other journals, and I'm thinking of journals like the MDPI and Frontiers and so on, they're vastly going up. So we need to push this line up with all our great society journals. Now, that's my little plug for the society there. And as we move on to the sort of recent years, we now know that bacterial communications and signals are incredibly diverse, both chemically and in the systems that are making the molecules and actually sensing those molecules. And I'm going to spend the next 5 hours going through each of those systems for you. No, I'm not. I'm just going to highlight one new finding that I think is really very interesting. Acyl homoserine lactones you know about. This is a new paper that came out in Nature about a year ago. They're a whole family of N-acyl cyclolysines that are in funny bacteria called the Bacteriodota, most of which I've never heard of, except for these bacteria are present in marine environments, like we had in our systems, but also in human microbiomes, like the oral microbiome. So bacteria like Prevotella and Porphyromonas have the synthases, response regulators, and they mostly, it's not really quite clear what they regulate, apart from a number of non-ribosomal peptide synthases. Oh, okay. This way. Oh, move the microphone, okay. Sorry, thank you. Okay, so we now know that quorum sensing is, or cell-cell communication is widespread in bacteria. We know there's a diversity of systems, and we can see that if we go back, there is this term called quorum sensing that stuck. It was coined by a lawyer, by a relative of one of the authors of this well-known review that introduced the term, where the idea was that a few bacteria make a little bit of signal molecule. As they grow, the numbers increase. When the concentration of the signal molecule and the numbers are high enough, i.e. they reach the quorate state, bang, on comes the target genes. Of course, we now know that the quorum is not a fixed number of bacteria. It's all to do with the threshold concentration of the signal molecule. And that signal molecule concentration will change according to the numbers of bacteria and the environment that it's in. So the rate of production and loss of that signal molecule will very much determine the number of bacteria you need to become quorate. So even a single bacterium like Staph aureus in an endosome can become quorum, can switch on its quorum sensing system because it accumulates that signal molecule in a closed space. So what we now know and appreciate is that recognition and communication in the unicellular world, unlike Jacques' thoughts, is actually pretty common. Whether you want to protect your niche, whether you want to combat host defenses, you want to produce lots of virulence determinants, you want to coordinate that, build a biofilm, or if things get bad in your niche, you want to escape, signaling to your mates, it's time to escape, is a good thing to do. And just to say, in terms of defining quorum sensing molecules, we can say that it's really a molecule that does accumulate extracellularly. It's recognized by a specific receptor, induces a coordinate response. The response should extend beyond the metabolism of the signal, and it should benefit both the sender and the receiver. And of course, what we also now know is that the whole population doesn't switch on together because there's a division of labor, and we see population heterogeneity. Some bacteria stay out of becoming part of the quorum. Okay, so what I want to kind of go through as the main part of my talk is an organism that I really enjoy working on, and that's Pseudomonas aeruginosa, which has a large genome for a bacterium, over 5,500 open reading frames. About 10% of the genome is devoted to regulation. And there's this very nice paper by Balsabramian showing the network of regulators in Pseudomonas. Is probably a little bit out of date now because there are so many more regulators known. But the quorum sensing system is all down here. So let's have a look at that in a sort of an anatomy of a sophisticated quorum sensing system where you've got multiple signal molecules involved. So in Pseudomonas, we've got a long-chain homoserine lactone, a C12 compound. We've got a short one, a C4, and we've got a couple of quinolones, two alkyl quinolones called HHQ and PQS. And what we know is that we have the LASAR system, which uses the 3-oxy-C12, the REL system, which uses the C4, and the PQS system, which uses these alkyl quinolones. And they interact with each other, and there's some hierarchy, but it's not absolute. And what the system regulates is a whole variety of secondary metabolites, biofilm development, swarming, exoenzymes and exotoxins, and antibiotic efflux. So lots and lots of genes regulated by this system that play an important part, particularly in the disease or infection-causing attributes of Pseudomonas. So I'm going to focus on the quinolones particularly. As a family of molecules, these are very well known. Quinine and chloroquine are two antimalarials that have been known for more than 100 years. Nalidixic acid was the first first synthetic antibiotic, first quinolone. Ciprofloxacin, of course, is one of the furanones that we now have. Now, Pseudomonas produces an alkyl quinolone zoo, and this has been known since the 1940s when the structures of these compounds were first elucidated. So we have compounds which don't have an OH, like we call the HHQ series, the PQS series has a 3OH, the HQNO has an N oxide, and then there's a molecule that lacks the side chain. So this is really a zoo, a soup of molecules that are made. So I'll just take you through a few things. How are they synthesized and sensed? What are their functions? Are they produced during human infections? Do they have potential as biomarkers? Can they be exploited as an antibacterial target? So the synthesis of these alkyl quinolones is a bit more sophisticated than the single Lux-I type gene that you need for the acyl homocysteine lactones. And you can see here that you have basically 10 genes. You start with anthranilate, and you basically add fatty acids and close up the rings. So you've got CoA ligases, Fab-H, you've got a thioesterase, a couple of monooxygenases that are important for putting in oxygen, and you've got PQSR up here, which is the transcriptional regulator. And if you put this in the context of the organism, basically both of these molecules can activate PQSR to drive up the synthesis of lots more quinolones. HHQ is the precursor of PQS, one of the monooxygenases, sorts that reaction out, and PQSL takes a precursor from the biosynthetic machinery to make the N oxide, and all these molecules are kicked out. If the environment changes, the organism can shut down making these alkyl quinolones and instead make two other molecules. A molecule dihydroxyquinolone, which we don't really have a function for, but it lacks an alkyl side chain, and 2-aminoacetophenone. If you ever smell Pseudomonas, that grape-like smell that it has is this molecule here, which has some immunomodulatory properties. So if you knock out A, B, C, D, or PQSR, they're all alkyl quinolone negative, but PQSE mutants can produce alkyl quinolone still, even though it appears to be a key reaction, a key thioesterase reaction. That's because there's another enzyme in the organism that can do this job. So if we take PQSR for starters, this is the response regulator. It's an LTTR-type regulator with a helix-turn-helix domain and a co-inducer binding domain in its C-terminal. And it basically works that there are two dimers that sit on a regulatory and an activation binding site. Oligomerization of the two dimers causes DNA bending, but you need a signal molecule to activate the system so it can drive the RNA, interact with RNA polymerase to drive expression of the downstream target genes. So both PQS and HHQ, as I said before, are co-inducers for this, but we didn't know what the structure or the nature of the binding site was. And so we purified the CIBD binding site and crystallized it with this C9 molecule we call NHQ. It's just got 2 carbons longer than the C7 PQS, but it's as active as PQS. And you can see each monomer in this dimer has got a binding site. And if we look at those in more detail, you can see that basically we have 2 cavities, or 2 pockets. The A pocket binds the alkyl chain, and the B pocket actually takes that alkyl quinolone on board. The interesting thing, you can see it's all pretty white, which means these are all very hydrophobic amino acids. It's basically a greasy hole that these very hydrophobic molecules sit inside. So the A pocket can take only C9 and C7 chains if you want to activate it. So these molecules all share similar EC50s. If you go to C11 or C5 side chains, it doesn't work. Okay. So we wanted to know what each of these individual signal molecules and the PQS-E protein was up to. So to do that, we made some mutants. So Giordano Rampioni in our lab spent a lot of time constructing a mutant which couldn't make any quinolones and lacked the monooxygenases. So when you fed things to it, it didn't process them to make any of the molecules that required oxygenation. We then also knocked out a delta 5 mutant where we knocked out the R gene, PQSR. And then we fed them either HHQ, PQS, HQNO, or IPTG and said, from a transcriptome point of view, what do they actually do? So there are lots of genes, as you can see, activating and repressed, different numbers of genes controlled, some the same, some different. But the key take-home messages are this. HHQ only drives PQSR-dependent expression of PQS ABCD. The N-oxide HQNO doesn't influence the transcriptome. It turns out to be a cytochrome inhibitor. PQSR only targets the PQSA promoter. PQS-E regulates multiple virulence genes, which is kind of interesting. And also, most of the genes controlled by PQS didn't require PQSR. They were PQSR independent. So if you take PQS-E, this thioesterase, which is involved in alkyl-quinoline biosynthesis, paradoxically, it's not required, as I've said before. But it does regulate virulence in the absence of the quinolones. So if you perform this experiment, so here we've got a mouse acute wound infection model, and you have a look at the wild type. That's the kind of virulence that you've got. That's the survival of the mice. You knock out PQSA, so you make no quinolones. You've got an avirulent organism. But if you put PQSE on a plasmid, gosh, you've got your virulence back. So what is it that PQSE does? Now, we didn't work this out. It was worked out by some other groups. Now, you remember that Pseudomonas has this nice overlapping set of regulators using different signal molecules. And the key part of this cascade here is the REL system. And it turns out that PQSE is essential for folding and stabilizing REL-R so it can interact with the C4 signal molecule and drive expression of the REL target genes. So these systems really are closely interfaced. Okay, I also said that most of the genes controlled by PQS were via PQSR-independent pathways. It turns out that PQS is actually a ferric ion chelator. It makes these lovely ion complexes, 2 to 3, with iron. And it turns out because it's hydrophobic, it interacts with membranes, as well as binding iron, it also induces membrane vesicle formation. It can either hand the iron over to the siderophores pyoverdin and pyochelin, or it can interact with a protein called TSEF, which facilitates delivery of vesicle-associated iron. So we're beginning to see that these molecules that bacteria produce that we're thinking of as having specific signaling properties interacting with the response regulator, in fact, have, well, multifunctional properties. Now, you have to be careful with some of the papers because sometimes people put in millimolar to molar concentrations of these molecules. These should be active, if it's truly acting in this way, at low micromolar concentrations. But if you take HHQ, PQS, or HQNO, they all interact with signal producers, with polymicrobial communities, and with the host. 3-oxy-C12 undergoes a rearrangement reaction to form a tetramic acid that turns out to be very active against gram-positive bacteria. Now, I'll just digress for a moment onto this molecule, some work that we did with an immunologist, Dave Pritchard, at Nottingham some years ago. So we wondered whether, given it was produced by a pathogen, this 3-oxy-C12 molecule, whether or not it might have an impact on host defenses, on lymphocytes in particular. And what we found was that 3-oxy-C12 at low micromolar concentrations was capable of inhibiting ConA-driven proliferation of lymphocytes. So we went one step further and said, well, if it is anti-inflammatory, would it have an effect on the development of diabetes in the non-obese diabetic mouse? And the answer was yes. So these mice, after about 14 weeks since they're born, will start to develop diabetes. There'll be glucose in their urine. And their pancreases will have been attacked by macrophages, and they should be well infiltrated. So that's what you see in the islets of the pancreas in the untreated mice. In the treated mice with thrioxyC12, they were pretty clean and healthy. And if you measured urinary glucose as a cumulative diabetes index, what you see that the compound worked quite nicely, whereas 3,6 wasn't able to do anything better than the control. And if you extended the side chain a bit further, that didn't work either. So we actually set up a spin-out company to try and develop this, but we didn't really get very far because it turns out this molecule is incredibly pleiotropic and has a lot of different effects on the immune system in lots of ways which are both pro and anti-inflammatory. So it really wasn't a good drug candidate in the end. So just a couple of final things to take you through. First of all, can we exploit alkyl-quinolone-dependent quorum sensing? And can we think about how we might do this? Because Pseudomonas can cause infections in pretty much any body site. So are these molecules artifacts? Are they really produced during human infections? Do they have biomarker potential? And is it a viable antibacterial target? So I'm sure you're very familiar with cystic fibrosis, with those mucoid airways because of the CF mutation, who get chronically colonized with Pseudomonas. And so we did some studies with our clinical colleagues in 3 different CF centers because we wanted to answer some questions. Can we find quinolones in CF individuals who are colonized with Pseudomonas? And is there any possible way we could use that information to develop biomarkers and diagnostic assays? So we needed to have a look at all of the signal molecules that we knew about, not just the quinolones. And what we found is that we could detect them in sputum, plasma, and urine. So that means Pseudomonas clearly makes these molecules in vivo during infection. One of the interesting things is you don't find very much of the 2 lactones in anywhere other than sputum, where you have a little bit. But you find buckets of quinolones, in particular HHQ, which is the C7 molecule there. And this is, of course, using LCMS, which, of course, you can't use at a patient's bedside. So if you have a look in urine, for example, and you look for this molecule, you can see that a patient who has never had or never been colonized by Pseudomonas, you don't find the quinolones. If they're free as well, you don't find them. If they're currently free, that is, of Pseudomonas, but they may have had it before. Or if they're intermittent, you might find the odd patient with a level of this molecule. But chronically infected patients have a wide range of the signal molecule in their urine, and the controls were thankfully negative. We did one study where we looked at patients who were stable, then had an exacerbation of infection, and were then treated with antibiotics. And what you can see, each of these is a single patient, each of the colored lines. And you can see that the levels of, in this case, NHQ, which is the C9 congener of HHQ, these fall quite nicely as you come towards the end of an exacerbation. Even though there isn't much of a change in the numbers of Pseudomonas, you clearly see that they're becoming more virulent by producing more of their signal molecules to drive those virulence factors upwards. And one of the outcomes of all of this work is that my colleague, Miguel Camara, and the clinical colleagues at the local hospital have spun out a company called MIDX, which is developing a lateral flow test to use they've been developing antibodies for the lateral flow test to actually detect Pseudomonas in CF patients and elsewhere. Okay, so here we're thinking about diagnostics. What about therapeutics and prophylactics? There's been a lot of interest over the years in whether you could develop an antivirulence molecule by blocking quorum sensing that could be used as a therapeutic or as a prophylactic. Well, you've got plenty of options in quorum sensing for developing such inhibitors. You could go for the synthases, you could go for the molecule itself, or you could go for the receptors, the LuxRs, the PQSRs, etc. Or you could try and take out the molecule itself with an enzyme or with an antibody. So antibodies you develop for a diagnostic could actually potentially be useful as a therapeutic. So here's the PQS system, which controls virulence of biofilms. And we've got PQS as a good target molecule. We've got PQSR. We've got the enzymes in the cascade. And we've also got PQSE, this odd thioesterase that's also an adapter protein. If you take PQSR, for example, which I'll illustrate, this is the kind of data you get from a mouse lung infection model where clearly the PQSR mutant is much less virulent than the wild type. So what you need to do some developmental work here is you need a good source of chemicals. You either have to make those chemicals themselves, either find some natural products you could screen, look at synthetic libraries, structure-based design, or even FDA-approved drug libraries, because that's a shortcut, because it's something, a drug that's already been made and used in the clinic for some other disease state. So just a very simple way of starting this kind of work. Here you've got a molecule PQS with an EC50 of about 1.9 micromolar. So can you take that template, chemically play around with it? We also have the structure of PQSR, so we can see what fits in the active site, to come up with an inhibitor. So the first thing we got did what lots of med chemists do, is to put a chlorine in a bicyclic molecule like this. And what we did was end up with an activator which was 20 times better than PQS, so you really wouldn't want that. But if you take a wild-type P. originosa, where the PQS genes are regulated by PQSR, and stick into a neutral site in the chromosome, the PQSA promoter, and Lux or GFP, you can have a nice assay for inhibition. So you can find out what kind of IC50 your molecule that you think might be an inhibitor would give you. What you're looking for is nanomolar levels of activity. So this is the molecule that I'll not tell you how many molecules we actually screened and synthesized. That we synthesized, which was the most promising initially, that gave us a nice lead. That had an IC50 of 4 micromolar, and you can see that if you give this compound to Pseudomonas, you can knock out, so green is the wild type, and then the red is the wild type plus the quinazolinone, which is what these molecules are called. And you can see that we can knock that out. We can knock out, with the quinazolinone, the production of pyocyanin, the classic blue-green pigment of Pseudomonas. And if we then look at the interaction between these fluorinated molecules within the active site, we could see that there was a very nice interaction with the threonine at 265 in the B pocket with the chlorine that we'd stuck on to this position here that was in the inhibitors and, in fact, in the beta activator, but was obviously missing from the natural product. So we wondered whether we could take this on a bit further, thinking about polymicrobial situations, because in the CF lung, you're going to have often Staph aureus and other organisms there. So we modified the molecule a little bit more in this way. So we extended the side chain here. So it's an alkyl amino side chain with about 4 extra carbons, and got a molecule with a reasonable IC50 of inhibiting the quinolone system in Pseudomonas. But what it did also do, it was very potent. It was quite antibacterial towards gram-positives and Staph in particular. So we thought this could be a useful way to deal with Pseudomonas biofilms. So in the last slide, no, the last section of the slide, let me explain this. So here we have a mixed Pseudomonas and Staph aureus biofilm. So we have Staph aureus in green, Pseudomonas in red, and the two merged up the top. If we use tobramycin to actually try and kill off these organisms, we certainly damp down the Pseudomonas. But doing that actually protects the Staph aureus, which is now growing much happily, much more than it was. But if we combine the tobramycin with the quinazolinone, this one at the top there, oops, I've gone the wrong way, this one at the top here, then you can see we've pretty much wiped out the biofilm altogether. Now, we've now probably screened several 100,000 plus molecules. The trouble with doing anything prophylactically or therapeutically in this field is the cost of taking it onto the next stages. So I'm going to actually switch from this and not take you through this any further, but actually introduce something that we've been doing for the last few years, which actually has led to some useful clinical outputs. But before I do that, I forgot I'd put this slide in. So this is to say that really a little bit about, is quorum sensing really a viable antibacterial target? The number of papers in the field has rocketed, as you can see, up to 2025. There's about 200 a year. There are lots of reasons why an anti-virulence agent might be quite useful, but I think we come to the conclusion, firstly, that you need to give it with an antibiotic, so you can kill the organism as well as reduce the virulence of the organism, and it might be better in a biofilm context. And also, a lot of these papers use something we call the PAINs. These are the pan-assay interference compounds. So things like curcumin, some of the herbal stuff, where there's no specific interaction. You can't develop a drug molecule for it. You can't modify the structure because it interacts with everything. You need a very specific drug-like molecule. So despite the huge amount of work that's been done in this field, there isn't really a novel quorum sensing inhibitor that's yet out there and being useful. So one of the things we've been doing is looking at biofilm-associated infections. And something that's come out in the biomaterials field is this idea of bioinstructive materials which can prevent medical device-associated infections, but also bioinstructive materials that can modify the response of the host, ranging from stem cells to lymphocytes, macrophages, and all those sorts of things. Now, let me introduce you to this character. You can't quite see it on there, but his name is Alfred Ridley. And he was the guy who was very keen. He was a surgeon who, working with patients with cataracts, came up with the idea of replacing the lens. He had a lot of opposition to it, and it wasn't until 1950 that he actually managed to get a lens into a patient that really worked. He got his idea from Spitfire pilots in the Second World War. So if you were piloting a Spitfire with a glass canopy, and you got shot up, you had bits of glass in your eyes, and your eyes became very badly inflamed. But if you had one with the new Perspex, your eye didn't become inflamed. So that gave him the idea. So we ended up with a Perspex lens. And of course, we now know that we use hundreds of $billions worth of medical devices that we implant into patients for all sorts of conditions. One of the main features as these have been developed, the main criteria has been that mechanical properties and chemical inertness. Really, infection wasn't really thought about. And of course, healthcare-associated infections, including implanted medical device-associated infections, cost the US between 35 and 88 billion and about 1 billion in the UK. And biofilms are the big problem. So one of my colleagues got me involved some years ago. He is a biomaterial scientist in thinking about high-throughput ways to find polymers you could use to coat or make medical devices that had good anti-biofilm-resistant properties and weren't dependent on the presence of an antibiotic or some other leachable material or leachable compound. So this is one of the stories. So what you can do is you can think about polymers in terms of their chemistry or in terms of their topology. And when it comes to topology, you can think about two things, either macrotopology or nanotopology, or microtopology, I'm sorry. So this little story is really about microtopography. So with Jan de Boer in Eindhoven, who developed this topo chip. So this has 176 to be precise, different patterns on it. It's been generated with a mathematical algorithm. It's made of things called primitives, which can be cubes, pyramids, or blocks, into different shapes, and then patterned out in different ways on the surfaces. So if you look down at the bottom here, you can see one of the patterns that's created and repeated. So what you can then do is to take this microchip and pop it into a culture vessel with your favorite pathogen, either labeled, or you can use a fluorescently labeled, or you can throw in a dye afterwards. And then you can look at what happens to its interaction with all 2,000 plus microtopologies. And you can generate these graphs. So right down the bottom here are the ones we're interested in, the ones where there's very little fluorescence. So the dark green ones we don't want, the black ones we do want. So just as an example, here's a flat, here's a resistant topology, and here's one that promotes biofilm formation. So you can see for pseudomonas and staph, both of them don't form biofilms. They don't like this particular surface. And if you implant 881, 697, or a flat of the same material into a mouse, into a model, you can see that it actually works reasonably well in a mouse. The question is, how does it work? So this is the 881 pattern. So you've got these funny features repeated throughout. But if you take your Pseudomonas in this case, and you do some video microscopy, and you have a look, I don't know how well you can see this from there, but we can see the bacteria tracking down the 881. And the turquoise color means they're moving quite quickly, so they're charging down it. That's within the first 2 minutes. Then, by the time you get to 2 hours, you can see you're getting these little groups of bacteria that are actually collecting in the clefts. But we know that you can't make a biofilm on this, so what's happening here? Remember this quorum sensing system. Is there a role for quorum sensing system? Somehow, working in reverse to prevent the organism forming a biofilm rather than promoting it. Now, one of the things Pseudomonas makes is a rhamnolipid. Rhamnolipids are basically lubricants. And if you put rhamnolipids on the surface or you give it to a biofilm, you can disperse them. Pseudomonas makes it and uses it to actually develop its biofilm, but it has to produce it at the right time. So what we know about rhamnolipids is that they're controlled by the C4-rel molecule, and rhamnolipids play an important role in swarming. If we knock out the rel-I gene, which is one of the rhamnolipid synthases, or you knock out rel-R or rel-I, which are the two genes, the R and the I, from the quorum sensing pathway, you can see that you don't swarm anymore. So rhamnolipids are needed to be able to allow Pseudomonas to move, so it means they're not sticking to a surface. So that might give you a clue. So what we found is that 881 could be colonized if we knocked out rhamnolipid production or we knocked out the rel quorum sensing system. So that suggests that early induction of quorum sensing, so bacteria accumulating in very small quorum, not large quorums, start making the rhamnolipid at the wrong time, far too early. That then covers the surface and they can't stick to it, so you don't get a biofilm. So basically, it's an auto-lubricating phenomenon that stops biofilm formation. So we're continuing to develop this particular group of topographies to see how far we can get with them clinically. We have done some other work on chemistries, and we have something very similar on a polymer we've now managed to get into the clinic. Now, I think that's probably all I'm going to have time to say for, because I'm sure we're probably almost at the end of time. So, although we now know, after about 35 years of work, an enormous amount about quorum sensing systems, there's an awful lot we don't know. There's an awful lot of questions that we still have, particularly in the context of polymicrobial interactions with the host. Why do these organisms make multiple signal molecules? What are the spatial and temporal dynamics in different environments? So basically, we can say the more that we learn about these systems, the more we realize that we don't really know that much. Now, I think I've talked for a long time about a lot of work. I've had to leave a lot of work out, and I'm also going to have to leave out a lot of people who've been involved in the work, but I would certainly like to highlight some of these guys here in work that I've mentioned. So Miguel Camara and Kim Hardy have worked with me for about 30 years. Stefan and Steve, who are both here, have also been with me for a long time and been involved in lots of this work. Steve Diggle, who's now in the States of Georgia, was with us also for a long time. Manuel did a lot of the work with Jean on the microtopographies I've just talked about, Giordano on the PQS circuits, Weng on a lot of the chemistry, and Morgan is the biomaterial science. And there are lots of folks here. I mentioned George earlier, in those early days where we had great fun figuring out what on earth was going on with the carbapenem system, with Ewinia, and with other microbes in terms of the homocysteine and lactones. And I certainly would like to acknowledge lots of the different organizations that have supported us over the years, and to say that there are many more people that aren't listed here who've been involved. And I really have appreciated the very many talented people that we've had in our labs who've really pushed this field on. Thank you very much for your listening. I hope that was fairly clear, and I'll hand back to Gordon.